Okay, so the next lab goes over Mendelian genetics. And mainly, I think what you're meant to get from this lab is just how to do a Punnett Square, how to see how things are gonna get passed on. And so one thing to remember when you're making a Punnett Square and why it's 4x4 is that those are representative of the haploid cells that we observed in meiosis. So let's do Freckles. Freckles is a dominant trait, so R dominant, little r recessive. And so that means if it's a dominant trait, that means you have freckles. If it's heterozygous, you're still gonna have freckles because you just need one gene to express this. But if you have recessive in both, no freckles. So remember, in the case of Mendelian genetics and what we're dealing with, if you have a dominant gene, one of those capital ones, or you're heterozygous, it doesn't matter, you just need one, you're gonna express that trait. So as we saw with the PTC taster, for example, let's say this is taster now. Let's see, these are just representing taster. These two people, on their cells, on their tongue cells, they have the little receptor because they have one gene that will express that protein receptor. These are functional genes, basically, the dominant ones are, and that means the dominants will express and you'll taste that bitter taste. People like me, though, that couldn't taste it, what recessive genes honestly usually are is that they're just mutated and you never have those receptors. So I lost that function to taste the bitter. So I taste no bitter. So that's just like something to think about when you're using and looking at these genes. That it kind of makes more sense when you know that recessive traits usually just mean nothing expresses, like no protein shows up. So back to the freckles. Let's say you have one parent that is heterozygous for freckles. And remember, in their cell, they're heterozygous. And another parent that's also heterozygous. So this is the classic example. Now the reason we use the square is, remember, when we do meiosis, there's a 50% shot that their gamete is going to be, that their little haploid is going to be the capital, like the large, the good gene, the big freckle gene, and half chance that it's going to be the little freckle gene. Same thing over here, big R, little r. So that's what Punnett squares represent. They're a representation of the chances of meiosis happening in certain ways. So now we can go over this Punnett square. Essentially, this is that classic example where you have two parents. Each one is going to split its little haploids, and the chances of a double dominant offspring are one up here. Chances of a recessive offspring, so you put here together, here, are two right here. And then the chances of a fully recessive offspring are down here. Okay, so one thing the lab also covered is ratios. So with Mendelian genetics, dominant versus recessive phenotype is all you're gonna need to know. So in this case, this one, and this one are dominant. They're expressing. They make up a 3 dominant to this one is not gonna express recessive phenotype, okay? But what you can see too, and this is, call this phenotype. So you can see that right here. These are all the dominant phenotype they're gonna express. The genotype ratio is different. So if you do a genotype ratio, you're gonna have 1 double dominant, you're gonna have 2 heterozygotes, and you're gonna have 1 little guy here. So just reading those Punnett squares, you can find your own ratios and make them. Now, you're likely not gonna see just a double heterozygote cross, because that's usually like the classic example. You might see something different, but be ready to be able to do a genotype ratio and a phenotype ratio. And in our case, phenotype is always just 1 dominant equals, that's the dominant phenotype. Okay, so take a pause here. If you have 2 parents that are, for example, if you're gonna do, say you have a parent that is just, we'll just call these some trait of some kind. We'll call this gene number 1 and gene number 2. So you're probably gonna see this where one parent, you're gonna do a big Punnett square for 2 parents. And let's say this parent over here is smaller, and it's also little here, let's just do it up here. So also a heterozygote for that one. So basically all you're gonna do for this one is split all the combinations of these genes into little haploids. So essentially, you're gonna do one here. So this parent, we're doing this parent first, and that's RA, capital A. Oh, shoot, let me just get that better. So you have to do this for each one. So that's capital, capital. You're gonna do it again here. You're gonna get another possible gamete set here. And you're gonna do the same thing for both R's here, and they're both gonna be on A, right? So this one will go here, right there. And this one will go here. For this parent right here, these are your 4 possible gametes now. Let's do this one. So for this one, little r, big A. Same thing, little r, big A here. Same thing now with the little a. Still gonna be little r though. Little a again, over here now. Good. And now you can just make up your giant Punnett square. It's easy to write it out. You have all of lab period to write this out, so feel free to do this to even just check your work. You know, we could ask you what are the chances of, you know, this genotype of offspring, and then this way you can just be like, okay, these chances are, you know, X based on the phenotypes and genotypes. So you can be able to, once you write this whole thing out, you should be able to just pick and choose. If you do want to, you can just do like, okay, so for example, what's the chance of A from this offspring, from these 2 parents, for example? What is the chance of a heterozygote offspring? So right here, you immediately see that all the little r's are going to combine. So if you do a mini square right here, this parent's over here, this is over here. Pause the video if you get slowed down or anything, and email me too if there's anything you want to know, like if I go too fast or something. You just do gene by gene, and then in this case, you see that each one is a heterozygote. So there's a 100% chance of heterozygotes for this R gene. Now you have to do A gene too. Oh yeah, and these are heterozygotes right here. So what are your chances of a heterozygote for the A gene? You have to do another Punnett square down here. And remember, you can just use this raw, but if you just want to do the quick math, you can do it this way. Double dominant, there's a heterozygote, and there's a heterozygote. Double recessive, 1, 2, versus all these. So this, one, when you combine, they're all gonna be that one. So that's just 1 times the chance of this is 50%. So that means that roughly 50% of the offspring will have this genotype. So you can do that, and then always remember the genotype can be different than phenotype with a dominance. Okay, and that should be good. Let me know if you have questions on this.